My interest is really in inflammation, and as trained as a pharmacist, to me it's kind of a question of drug delivery, and so we're going to talk about three concepts today related to targeting expression for imaging, and that will play out thusly. We're going to review some basic principles of SPECT and PET imaging, which I hope you're all familiar with, some of the requirements of the probes and the imaging systems, and also the facilities and personnel that you need for novel probes to make that translation, some study design considerations, and I'm really interested in the quantitation that is possible from imaging. So imaging as a sport can be very sexy, and radiologists certainly have a lot of experience bringing qualitative interpretation to imaging in the context of disease, but I want to turn this around and make it really more of a quantitative sport where we can describe the pharmacokinetics and pharmacodynamics of drug delivery using imaging. And then we're going to digress just briefly and talk about some of the clinical imaging resources that are available to image infection and inflammation, and why we need better, more specific imaging probes to ask and answer these questions. So all of what I'm going to talk about today relies on this sort of basic trilogy that, to use imaging successfully, whether it's clinically or preclinically, we have to have a very thorough understanding of a disease, and sometimes this manifests itself in animals, in a disease model, in nude mice or some transgenic mouse, for example. A probe that's well characterized, that we understand the ligand-substrate interaction, and then an imaging system that can gather the relevant information in both a spatial and temporal scale that's relevant to the question that we're asking. So really we have to think carefully about each of these three components before we can begin to ask and answer the important biological question. The technology itself has been around for about 50 years. This is basically an unsealed source of radioactivity that's injected into, in this case this is a human, and it circulates systemically, and we rely on some selective targeting or preferential metabolism and clearance to give us some localization of a probe. And in that sense, we create a high target to background or signal to noise concentration. This technology has been in the clinic since about 1970, but recently, we've had some advances in small animal imaging that I'll talk about in a moment, but essentially it's the same technology. This is 4 different planes of detection, very large, solid crystal type detectors. They acquire data in projections, and in this particular system, we're actually, on each head, we're acquiring 9 multiplexed, multiple images so that they can be brought together in a projection, and then they can be deconvolved, and through tomographic back projection reconstruction, we get a 3D model that we can go in and interrogate in any plane that we're interested in. That's just basic SPECT imaging. The real purpose of what we're trying to do is to bring the same resolution in a mouse that we can achieve in man. These are clinical studies that are sort of off the shelf at this point. This is DAT-SCAN that's recently approved, and quantitative left ventricular ejection fraction imaging. And these are very much the staple of clinical nuclear medicine, and the resolution we achieve is maybe on the 1 centimeter to 2 centimeter cubed kind of scale. Well, if you do the math and think about how big a person is, maybe 70, maybe some people more like 100 kilos, I don't know. To get the same resolution in a mouse that's 10 or 15 grams, we have to do quite a bit better, maybe 4,000 or 5,000 times better. And actually what we are able to achieve in this small animal imaging is sub-millimeter resolution, which now gets us into the realm where we can make that translation, and we can really compare what might be useful in rodents to what is achievable in man. And so the translation is much more linear. I wanted to give you some idea of why we think SPECT and PET has some unique capabilities to ask and answer these questions, and it really has to do with this probe sensitivity. This is this is related to the amount of radioactivity that we need, to the concentration of the probe, and we can do studies in biologic systems where we're really down to the femtomolar kind of concentration of probe, and that keeps us well below the mass action threshold, so we can stay below that amount of drug that might cause a pharmacodynamic change in the tissue. So this is very important for studying receptor kinetics, or really any in vivo concept without perturbing the in vivo milieu. And so that's a very important advantage of SPECT and PET over some of the other modalities. MRI, we call that a millimolar sport, and nuclear medicine is at least a nanomolar and probably more like a femtomolar, then we have to also think about the depth of penetration. There's a lot of good techniques available in fluorescence and bioluminescence that are particularly useful at the cellular and tissue level, but when you get to the whole organism and you start thinking about imaging disease in situ, it gets very difficult because of the attenuation of the optical photons by the overlying tissues. So there are times when you can do intraluminal imaging of in situ disease, but in general, you're really limited to subcutaneous and very close to the surface imaging paradigms for optical using fluorescence and bioluminescence. Then we have to think about this temporal window also. If we're interested in measuring kinetics and interrogating a system, PET radio tracers have some unique advantages. If my pointer's not working anymore. But really in our mind, we're interested in a snapshot of some expression signal at any point in time, and it's not so important that we're going to challenge and re-challenge the system. And because PET has such complicated chemistry in terms of incorporating halogens into otherwise candidate molecules, it's very complex organic synthetic chemistry. We think there are some advantages to SPECT, and then also you can follow these SPECT tracers much longer over time. So if you're interested in metabolism or downstream signaling, there are advantages. So I'm not saying SPECT is better than PET, but you have to choose carefully and think about what it is you're really trying to ask and answer. In every case, we're taking advantage of the concept that we need a ligand, the biologically active compound that can target some unique expression in the body, and we have to add the radioisotope that essentially becomes a signaling atom. And so we have to put those together in a way that they stay together, that we can characterize their fate in vivo, and oftentimes this involves some chemistry by chelating metals, or in the case of PET, actually using nuclidic substitution to put the fluorine in the place of a carbon, for example. We do a lot of this in our facility. I'm just kind of showing you some of the support that's necessary to do the radiochemistry, the radiolabeling, the formulations, and to characterize the metabolism and biological activity of these compounds. That's a pretty serious sport in our world. We do everything from simple one-off animal studies all the way up to GMP clinical studies. We've done 11 compounds under CGMPs. We've been audited 3 times by pharma companies, and we're a registered FDA testing facility. That's kind of an onerous burden to maintain all of that capability, but we have it to do these sort of boutique phase 0 to phase 1 studies. Once we get into a phase 2, 3, then that's really beyond the scope of what we can do internally. But our ambition is really to do these first in man and to identify biomarkers that have the capability of providing unique insights in human disease. Most of the time we have to start thinking about the studies in terms of the tracer, the animal model, and the imaging system, and I just kind of wanted to show you that there's different complexities. Oftentimes there's off-the-shelf tracers, some of which are used in clinical medicine, that can be very useful to inform about what is happening in an animal model, and it's really just understanding how it fits into your question. You can generate a lot of data very quickly, and when you make this more quantitative, it gets a lot more complicated. So this data analysis and reporting is at least as difficult as generating all the data. So just a word of caution that we realized early on that we have a high throughput imaging capabilities, and we can image 10 or 15 animals every day, but every animal has 20 compartments and different tissues, and if we're interested in getting quantitative data from those compartments, then it gets to be quite a challenge to wrestle all that data down and do cohort analysis. We aligned our resources very close together in terms of the lab so that we could go back and forth between SPECT and PET, and that creates some problem with radioactivity, and so we have to have a shielding wall to shield the SPECT camera from the high energy photons of PET. We have to have the right people to run these cameras and to analyze the data, to work with the animals, to do the type of animal procedures we need. We have BSL-3 capabilities in this lab to work with things like botulinum toxin and H1N1 flu and things that people are interested in looking at animal exposures. This is just a little picture of sort of the scale of what we're working with. This slide I included just to show that There's a wide variety of radionuclides that we can use in single photon emission computed tomography, or SPECT. And some of them can even be off the shelf. If you pick up a Aldrich catalog or Fluke or something, start looking at I-125 labeled compounds, there's scads of them, millions of them. The real question is whether the specific activity can be useful for in vivo experiments. Oftentimes, they're used for RIA or some cellular-based assay, and so the mass that you need to get enough radioactivity to generate a picture is insufficient in the specific activity that's available. But we do make pictures, and I just wanted to show you some images of showing a mouse and a rat, and these are relative scale. So that's a big mouse. That's a 30-gram mouse, biggest mouse I've ever seen, and almost 400-gram rat. And this is kind of the relative scale. The gray scale is the CT image, and then the color scale is our functional tracer that we're interested in. And these probes have different half-lives, and we can combine these probes and do multiple energy discrimination in SPECT. And this is part of the unique capability of SPECT, that we can look at multiple probes simultaneously. And I think you'll hear a little bit later how Aaron has harnessed some of that capability to look at some unique activity of cell signaling and immune cell recruitment. But again, this is a grayscale image of CT with a bone tracer. That's the color, the purplish color, and the green is just a sodium iodide distribution. And you can see there's a lot in the thyroid and the gut. And we can bring all these images together because they're acquired simultaneously while the animal is under anesthesia during one session. So there's no need for fiducial markers or complex co-registration or repositioning. This really happens simultaneously, so the data upon acquisition is co-registered. We've done some work with our collaborators at Erasmus University in Rotterdam to show that in vivo imaging data correlates very well with the actual tissue distribution, and this is just looking at some isolated kidneys. We imaged the kidneys, drew a region of interest around the kidneys, harvested the kidney at necropsy, and then counted the kidney ex vivo, and you can see that the correlation between in vivo and ex vivo is very good. But we don't want to have to do that every time we make an image. The purpose of doing the imaging study is to not sacrifice the animal, so we can do longitudinal observations, and we can look at multiple time points post-intervention or throughout the progression of a disease. And so we'd really like to be able to draw regions of interest around the animal and directly quantify, and in this way bring absolute quantification to this data set. We think this is pretty easy now that we've been doing it for 4 or 5 years, but we still haven't moved the field to the fundamental units of molecular imaging, which I believe are moles per volume per time. People still talk a lot about percent injected dose per gram, and that's informative, it's meaningful, but really to put us in the parlance of the pharma industry, they want to know how many moles of a drug are in a space over some given amount of time. And we can do this by being very careful in terms of understanding the dose that's administered and the percent ID per gram, but then also understanding the characteristics of our imaging apparatus, what the response is, or the intrinsic efficiency of radioactive counts per injected becquerel of activity, and then knowing how much specific activity, or becquerel per mole, or millicurie per microgram, whatever the unit is for our probe. And in that way, by making this relation between the dose injected, the radioactivity per mass, and the response of the camera, we can very easily get to mole per volume per time. Okay, so I'm going to digress for just a minute about the clinical tracers that are available and why this is an opportunity for molecular imaging. Infection and inflammation in a clinical sense is imaged using tracers that are largely non-specific, and the most frequently used is just a bone scan, where we inject the patient with the bone-seeking radiotracer. We take an immediate image of blood flow to look at perfusion. They take a delayed blood pool image to look for extravascular leaking, and then they take a delayed image to look at the actual bone uptake or osseous phase. And if you have positive radiotracer accumulation, target to background in each of these 3 phases, that's consistent with an infection. Remember the hallmarks of inflammation: Ruber tumor, Dollar, Calor. So we have hyperemia, we have leaky vascular elements that allow the tracer to get into the extravascular space, and then we have bone uptake because there's actually bone remodeling. That's good, but we can actually have problems with bone scans because of RSD, which this conference is about, and other neuropathic disorders that can actually affect the bone. So we sometimes add other tracers that bring a different functional aspect to that probe. And in this case, I've shown you some examples of gallium with the bone-seeking radio tracer. But again, with gallium and a bone-seeking tracer, you can have concordance, where if the gallium uptake is more significant than the bone uptake, people would interpret that as consistent with inflammation and infection. If it doesn't show more gallium than bone scan, they would think that's negative, that the bone is reactive, but it's not consistent with an infection or inflammation component. Or if it's equivocal, that they can't really say that it's spatially congruent and that the intensity is more with the gallium. So despite these multiple modalities, we still have a lot of problems with the diagnostic accuracy. We've used radiolabeled leukocytes since about 1978 clinically, maybe for another 15 years preclinically before that. But this relies on using autologous leukocytes, and the big problem is that in humans, the spleen is a dose-limiting organ. A lot of leukocytes traffic through the spleen, and especially when you take them ex vivo and go through these complex manipulations and damage the cells and centrifuge them and then re-inject them into the patient, you really can't rely on them trafficking as a normal autologous leukocyte. So there's a lot of limitations that we found for leukocytes. And here's an example of an HMPAO labeled leukocyte. You might remember HMPAO. It's a brain tracer. It's a lipophilic oxime derivative, but it also crosses cell membranes fairly readily, and we can label autologous leukocytes, but it's not very good for central indications because a lot of it ends up non-specifically distributed to the gut. But for bone, as in this case with a hip replacement, some hardware, it can be fairly useful. Sometimes we want to add these together again using the bone scan method, paint in some background, improve our target to background signal. So we have the bone-seeking radio tracer on the right, showing a vertebral hotspot. However, with the white blood cells, there's no vertebral hotspot. So then where does this leave us? We're not sure. FDG has gotten a lot of attention in the last few years. It's the molecule of the century in nuclear medicine for the 20th century. Everybody likes FDG because it can do a lot of different things. And in the end, it's a metabolic tracer. So we have to think about its tissue distribution is really a reflection of the tissue metabolism. It's not necessarily specific to any disease unless that disease is related to metabolism. So in myocardial ischemia, for example, that might be important. But in terms of imaging osteomyelitis or inflammation, it's very nonspecific. This is a case where we've added FDG with MRI to increase the specificity, and MRI, of course, is such a great tool for giving us high-resolution anatomical images, and when we bring this together with PET, now we get both high-resolution functional and high-resolution anatomical images, and this is kind of a slam dunk. Yeah, this is not Charcot, this is osteomyelitis. If we compare all these modalities in an inflammation and infection setting, you can see that the sensitivity is generally very high, but the specificity is all over the board. And as a single entity, bone scan specificity is terribly low. Even FDG, PET, it's 90%, but there's an incredible confounder for any type of mechanical injury or surgery or even an aspiration to draw a fluid sample from what is thought to be a diabetic foot causes tremendous problems with confounders because it creates a hyperemic condition where the tracer is deposited other than by its signal for metabolism. So we really need a molecular imaging probe. And I think that there's multiple pathways that we could envision for targeting molecular imaging probes, and I'm going to talk a little bit about why I think leukocyte function-associated antigen 1 is important. But there are several drugs in the pipeline that are targeted at influencing the role of selectins and cytokines, chemokines in inflammation. Certainly COX-2 inhibitors are the classic model, and people have tried to harness that selective expression for imaging. Right now there's some VLA4 targeted compounds that are trying to influence the reaction with VCAM1. But I wanted to talk a little bit about LFA1 and why we think that that's important. So here's an example of from the literature just last month where fucoidan is a P-selectin targeted compound, and they've done a couple different models. On the left is a ischemia reperfusion model, and you can see in the myocardium that there's intense uptake. It's pretty global. We can't really make out individual vessels. That's maybe a limitation of the imaging device, but they did do some autoradiography, and they did correlate that, in fact, the P-selectin signal that they find on the radiograph correlates with the inflammatory response by immunohistochemistry. And that's the general concept that we try to use in our lab is we look for imaging findings, but then we have to have tissue correlates. They did the same Fucoidan imaging in an infective endocarditis model, and I don't know why they flipped this image backwards, but maybe that's just to make it beautiful. But you can see in this image, what I want you to appreciate is, yes, there's uptake in the heart, and that's correlated by the, looking at the vegetation in immunohistochemistry. But look at the rest of this animal, and particularly in the spinal column, there's a lot of uptake of this fucoidan. And I'm not sure why that is, but this is a platelet-rich thrombus model in an infective endocarditis, and I don't think that probably affects the spinal column. So this P-selectin expression is either fluid in those tissues, or they've got a model that doesn't allow them to discriminate from that. So as usual in an imaging article, they don't have a lot of molecular mechanisms to explain that. So as I mentioned, we're interested in LFA1 and its role to slow down leukocytes and allow their movement from the endovasculature into the surrounding tissues. And LFA1 itself is expressed on leukocytes, and it's not expressed on any other normal tissues. So it's a very restricted expression in these leukocytes. It's a heterodimer of alpha-1 and beta-2 integrins, and it has actually 3 conformational forms that have been isolated, sort of a low affinity, medium affinity, and high affinity. As that I domain unfolds, it gets more active. And that unfolding of the I domain is actually what allows the interaction with intracellular adhesion molecule 1 and the stickiness and the slowing down of the rolling leukocytes in the endothelium. There's both inside out and outside in signaling that happens through this activation of LFA1. It works on APC interactions, and it also increases IL-2 and MAPK expression. So just to drill down on this for a second, this interaction with ICAM1 acts as sort of a tether, so that as these leukocytes are moving through the vasculature and they activate their LFA1 arm, that starts to increase the number of interactions. There's many LFA receptors expressed on the leukocyte. This is just showing one interaction with ICAM1, but the more activated it gets, the more expression we have, the more likely it is to interact and actually then diapodese and move into the endothelium and become a macrophage. So we stumbled upon this small molecule that was a castaway from Boehringer Ingelheim that was originally sort of a putative antagonist of LFA1 expression as an intervention in asthma. And when it didn't pan out to have the effects that they desired, it went back into the bin and became sort of an orphaned compound. And this small molecule did show extraordinary specificity for LFA1 expression, so we thought, well, maybe it could be a molecular imaging probe, and if we put a linker on it and an appropriate chelator, we essentially doubled the molecular weight. It's a very small compound. And the real trick is that it's only one stereoisomer of the LFA1 that actually does the kinds of things that we want to do, and it's very difficult to isolate that stereoisomer, and in fact that became the subject of a patent that brought a lot of value to that small molecule that Boehringer Ingelheim didn't have the technology really to do. But with Seprecor and the possibility of actually separating individual isomers, we have tremendous value now. We start by confirming that this is a species-relevant phenomena by looking at a fluorophore in flow cytometry in human lymphocytes and also in murine T-cell lymphoma cells to characterize the cell binding association constants, and it's around 100 to 200 nanomolars. That's not great for nuclear medicine, but because of the extremely restricted expression of LFA1, it's really quite good enough. So we showed that it's relevant in both human and murine LFA1 expressing cell lines. We characterize the molecule as in the radiolabeled form through mass spect and different radiochemical purity and radionuclidic purity tests to show that we can incorporate radionuclides with near quantitative yields and that we get a single species of the radiolabeled small molecule, and we can confirm the conformational structure. Then we have to go back and recapitulate that the findings we found from the fluorophore in flow cytometry can be repeated using our radiolabeled conjugate, and in fact we get similar cell binding affinities, and we know that the EC50 is about 50% of the KD, and we measure the KD at 286, which is pretty close to 183. Maybe not for the purists, but close enough for our models. A couple more slides about the in vivo imaging experience. This was actually done in a lymphoma model. We were thinking that this might be a targeted therapy for lymphoma. And in this ESBL tumor model in DBA2 mice on the left, these cells were injected via the tail vein, so it's a systemic expression of lymphoma. And when I first saw these images, I was nonplussed, to say the least, but really quite discouraged because it didn't show any focal uptake. And the more I started thinking about the distribution, you can see there's a small hotspot in the distal ileum here. I guess we don't have a pointer. This is this lymph-associated gut tissue that's in the terminal ileum. And so we confirmed that, in fact, we're targeting lymph tissue rich in leukocytes and this ESBL cells circulating through the body, probably some uptake in the liver. And when we looked at a normal animal, we didn't see any of that. So we confirmed that we were able to target the ESBL, but it's just a bad model for lymphoma. And again, we saw this very high uptake in the terminal gut. And when we look at normal Black 57 mice, there's really very little that stays in the blood, but a lot of it stayed in the tissues that have this signal. So looking at an inflammation model where we inject E. coli cells in the flank of a otherwise healthy mouse, and then there's a sham injection in the right flank with saline, we can see that there's a huge signal in the affected side compared to the contralateral as quickly as 5 minutes post-injection. And this is some of the unique characteristics of a small molecule, that they circulate and reach the target very quickly, and they tend to be cleared very quickly. And so really at every time point post-injection, we have a very large target to background signal to noise ratio, and we can image all the way out to 24 hours in this case. We've done some work characterizing the inflammatory response following H1N1 exposure. This is a ferret. On the left is the baseline ferret, and you can see there's quite a bit of uptake in the gallbladder. That's part of the normal distribution, but there's really nothing in the lungs. Ferrets are quite long, and they're all lung, all the way to the pelvis. And on the right panel, following the exposure, this is 1 hour post-injection in each case with the same relative amount of radioactivity, 7 days post-exposure to the new Caledonia 2099 H1N1, and yeah, I can't get it to, oh, there we go. And you can see the lung is showing a profound inflammatory response as evidenced by this localization of the LFA1 expression. So our conclusions from this experience is that molecular imaging probe demonstrates specificity for LFA1 in vivo, that we can image the recruitment and immune cell activation in a couple different disease models, including the exposures to toxins and infectious agents. So as a segue to Dr. Milligan's work, I don't want to steal her thunder, but we've done a little bit of work using older technology, radio-labeled autologous macrophages, peritoneally-derived macrophages, labeling with indium, a very non-specific kind of label, but it's a unique cell population, and she has a model for neuropathic pain with a chronic constriction injury in the nerve, and following this injury, we showed that the macrophages accumulate in the spinal cord and even have some recruitment into beyond the local dorsal root ganglia. And this correlates very well with the LFA1 expression by immunohistochemistry. And so our next work is going to focus on the LFA1 expression that we think is going to be a unique molecular signal for some of this inflammation that's caused by the chronic constriction injury. These are my opinions and may not be shared by others, but about 1968, the FDA decided they wanted to regulate radio pharmaceuticals, and so they moved from the Atomic Energy Agency at the time to the FDA, which means that they went and continue today to go through the full review that all drugs go through for safety, toxicity, and efficacy. And they do have some streamlined processes that diagnostic probes go through, but in general, it costs a lot of money to develop radio tracers. And so from a just return on investment strategy, most of the big pharma companies don't invest in radiopharmaceuticals because it's very unlikely you'll have a billion-dollar drug at the end of the pipeline. But there are smaller companies that now have been bought up by the big pharma companies like Bracco and Squibb and Eni and DuPont, and they're more part of big pharma. But in general, the return on investment has been difficult to realize, and the regulatory framework has been difficult to negotiate. There's been at least 3 molecular imaging probes that specifically target activated granulocytes approved by the FDA, labeled antibodies or fragments. Each of them, when they got to market, they had problems. The most recently, Lutech was a $100 million a year drug for imaging infection, but 4 patients died after receiving Lutech, and it was traced to some manufacturing problem, and the company that was manufacturing it decided that it was more economical to discontinue marketing of the drug than to fix the manufacturing problem and to reapply to the FDA for a new pre-market approval. So this is complicated, but there's some hope, and that is that Avid, was just acquired by Lilly for $800 million on the basis of AV45, which is a PIB-like compound for imaging the neurofibrin tangles and plaques that are associated with Alzheimer's. And Lilly, I believe, probably, I don't know if there's anybody from Lilly here, but you might ask them. I imagine before they spend $800 million, they probably have a strategy that diagnostic probe is going to enable some targeted therapeutic that they have, either as a staging or monitoring of therapy. So I think we're seeing a new dawn or some new reinvigoration of the interest in companion diagnostics, and particularly as it relates to personalized or molecular targeted medicine. We're not going to see a drug like Herceptin approved at the time on an 11% response rate. We're going to see a Herceptin trial that only enrolls patients with Herceptin expression throughout the trial that shows a 70 or 80% response rate in a very narrow and highly selective population. So I think drug companies are really rethinking how to use some of these molecular imaging probes to get drugs to market more quickly. Whether they become standalone diagnostic radiopharmaceuticals or not is still a different question. But there's a lot more interest from pharma, and that's really what we spend 90% of our time in the lab doing pharma studies to characterize PK/PD of molecular targeted compounds. We have a lot of excess capacity for imaging, and that's really what we do day and night, ask and answer those questions for pharma.